Hi there, and welcome to the third lecture of this series. So, you all know about bioinformatics, and if you have a bioinformatics background, I think you should be familiar with NCBI. So, NCBI is the National Center for Bioinformatics and Biotechnology Information. So, when we look at the NCBI, we know that contains a lot of databases, sub-databases as well, where we can download different type of data, but you can also access NCBI using E-Utilities, which is intra-abstract on Unix command line terminal. So it is called eDirect. So eDirect provides access to NCBI suite of interconnected databases, for example, publication, sequence, structure, gene, variation, expression from a Unix terminal window. So for example, you can search for any type of data on the terminal as well, and it will automatically download the data for you. So individual operations can connect it with the Unix pipe as well to construct multi-step queries, which is pipeline creation. So first of all, we have to install the eDirect onto our Linux system and Macintosh system. If you are following this tutorial, you can do this on Mac as well. So what we need to do here is we need to create a repository called Bioinformatics. You can create it by any name, but I am going to go with the Bioinformatics. So I have already created Bioinformatics directory in the recent lecture. So I'm just gonna go in it and see if there are any kind of file. No, there are not any kind of file that are available. So I'm just going to copy this command. So if you are using the terminal for your first time, you wouldn't have a curl installed into your system. You can install that, or you can just go with the wget command and paste it in your terminal and then press enter. After pressing enter, it will take some time to install, but once it is installed, it will show you that you can use the eDirect onto your system. So while this is installing, I already have installed in my system because I use a lot of bioinformatics tools, and I use this for my sequence coverage and downloading different types of sequence as well. So eDirect will run on our computers under the CY Unix emulation environment, and also can run on the Windows PCs as well. So to install the eDirect software, open a terminal window and execute one of the following two commands. So we already did that, and now IntransDirect has been successfully downloaded and installed. So how we can see it is installed? So we can use the command eDirect. So let's just go with that. So this will download the script and compile it into the eDirect folder directory. As a convenient one install, you have to run this command. So we will run this command and then we will press enter, and now eDirect is installed in our system. So there is a quick readme guide if you want to go use to. You can also search for intras programming. You can see the contents of this book as well. So there is 1 hour reading time on the book. So I have filtered out some information that we need in my documents, which I will upload to a GitHub library for you guys. So installing the e-utilities, you can use this command. For running it, you have to export it to the path. After that, you can have programmatic access to the programs that you need. So there are 4 types of program that we are needed. So for example, we first need esearch to search something in the databases. After that, we need the e-link to interconnect the databases. And if we want to filter out any kind of information, we can use the efilter command. After that, we can also use the efetch to fetch any kind of data from the databases. So these navigation program communicate by means of small structured message, which can be passed between the different operations by using a Unix pipe. So what is a Unix pipe? This symbol here, it's called a pipe. For example, I want to use esearch tab. For example, if I use this, so the esearch db protein and the database, and now it is interconnected using a pipe. For example, I'm going to use this example that was giving in the script, and I am going to use another command by piping it efetch faster dot txt, and now it will download the files that I need. So I'm just gonna cancel that, and let's get to our tutorial. So if it doesn't give any argument at the first, you can also go with the help app. So if we need help, just like I showed you with the esearch, there are e-link help. You can use the e-link to um, look up what it is does, so why it is related to, and how you can get any type of data using the e-link as well. So you can mess around with these type of programs. I will provide you this file, and you can also construct multi step queries. So just like I told you, eDirect allows individual operation to be described separately. Combining them into multi-step query, you will need a vertical bar which is called Unix pipe symbol. So you need a Unix pipe symbol. For example, if we copy paste this command, and now I'm gonna get you through the command. So esearch is a function that we are using, and it is searching in the database which is /db. PubMed is the database that we are searching. So what is the query we are using? /query TN3 transposition immunity. So all the articles related to this will be fetched, and the efetch system is giving us the function that is the format, which format it should be giving. So it will be giving in the medline format. So the results of this data depends upon how much faster internet you got. So my internet is kind of slow right now, so it is taking a lot of time to generate some files. So but now it is searching. So here we can see the result is generated. All the data related to the file we were searching about is now generated. So our query was TN3 immunity. So structure and function of plasmid and related plasmid. Here we can see all the data is present. Their PMID, own ID, medline, related article, and this is how you can create Unix pipes. So there is how to access some accessory program. So enquire is a function that retrieves the data from a remote server with the URL constructed from command line. For example, you need the data from an other database, but it is connected to PubMed as well, connected to some other databases of NCBI as well. So you can use enquire. So let's just copy this command and show you guys how this works. So let's just say I'm gonna enter this database. So it is going to show us the PMIDs of research articles that are associated with this transmitted pattern data element. So this is how this works. So you can also discover by navigation. So PubMed related articles are calculated by statistical text that follows the retrieval of algorithm using the title, abstract, medical subjects. So the connection between the papers can be used for making discoveries. As example of this finding, the last enzymatic step in the vitamin A biosynthetic pathway, we are going to search some articles about it. For that, we are going to execute this command that I have written for you guys. So here we can see if we run this command, there is a function esearch that I already told you. PubMed, the query is given. E-link, it is related to other databases as well. And the target is the find the protein, and e-filter, the organism we are selecting is the mouse, and the source will be the RefSec database, and we are going to format the fetch the data in the format of FASTA. So this will now execute, and as this is executing, let's just move to number 7, sequence reports. So let's just wait while this completes. So you can use this terminal and bash to download all kinds of data that is related. So I have made up some example queries for you guys, but you can use these queries to create your own queries, and you can download the data according to your own form. If you need any help with this, or you want to create some other files, you want to create some databases, you want to create something like that, so you can just always ask away, and I will try to give you each one of you the answer of that. So just for making the change, you just have to change the query. So like I'm searching for lycopene cyclase, you can search for P53 pathways. You can search for any other thing. You can search for the nucleotide data, not the protein databases. You can change the organism according to your own need. My internet is not working, kind of not working right now. I think so. So this is taking some time. So while this is working, I'm going to move to the retrieving the sequencing report. Nucleotide and protein records can be downloaded in the FASTA format. For example, if we use the esearch to search in the database of protein, and the query will be again the lycopene cyclase, and fetch the data in the format of FASTA, will it generate some results? Let's see. If we open another terminal tab and paste our query, let's just wait if it is working. So it is working right now because it will generate some files. So here you can see, I'm just going to cancel this. Here you are seeing a lycopene cyclase domain containing protein from Herbula species G356, given us the protein sequence. So just like this, we can also obtain the records from the GenBank as well. So let's see, if we paste this data, and we are searching the protein, query is lycopene, and format the format that we are choosing is GenBank format. So let's enter this query and wait how it returns the data, because we have changed the database format. First it was FASTA file, now it is GenBank format, and let's wait how it generates the query results. I think there is some issue with my internet because the files are not generating, and they should have been generated by now. And if we are going to search again, if I can show you guys, that's a serious issue. So anyways, you can try this on your own, because I will give you the file onto my GitHub repository, which is going to be named at Bash for Bioinformatics, and I will include this as lecture number 3, and you can use this to uh, download the data and go with your own research. And there is something that we have left as well. So there is searching and filtering option as well. So the refined results that you contain, the current result that you generate, can be refined by searching in the intervals, useful in the protein database for limiting the BLAST neighbors. I'm also going to show you how to use BLAST in the command line as well. So that is going to be our next lecture. So while this is happening, I'm going to just show you. So here you can see, uh, I'm sorry for this. Gonna close this, and our data that we given is now generated in the GenBank format. So here you can see the sequence is here, the locus tag is there, and you can use this as well. So now we were moving on to filtering some queries. So we are going to search for the PubMed query will be opsin gene conversion, and there will be link to related databases, and we are going to filter the query tracheotomy, and now it will generate some reports. So now we are doing the filtering of things, so we can also use the e-filter as well. So that's all for this lecture. I will upload this file so you guys can have it as well. And now you can see it, internet is currently not working, so it is generating some kinds of error, but the command is working. You guys can try out yourselves, and if you guys ran into any issue, do let me know. And uh, that's enough for this lecture. I think in the next lecture we will continue using the command line bash, and we'll get install the blast program from NCBI, and we'll use it to blast some species. And that's it for this